Oxidative phosphorylation in uncoupled mitochondria. Oxidative phosphorylation is the process that ATP synthase utilizes the proton gradient created by electron transport chain to produce ATP, and in this process we consume oxygen. That's why it's called oxidative phosphorylation. However, in mitochondria from brown adipocytes, the presence of UCP1, also called uncoupling protein 1, in the inner mitochondrial membrane adds another layer of complexity when it comes to oxidative phosphorylation. When activated, UCP1 increases the proton conductance, moving protons from the intermembrane space into the mitochondrial matrix. And as a result, the mitochondrial membrane potential decreases upon activation of UCP1. And the activation of UCP1 is very important for the adaptive thermogenesis, which means the capacity of the body to generate heat without shivering. And this process is very important for small mammals to keep their body temperature, but also activation of thermogenesis, or at least activation of UCP1, has been proposed as a tool to combat metabolic diseases, for instance, obesity. So for a very long time, it was believed that oxidative phosphorylation in brown adipocytes, or mitochondria from brown adipocytes, was irrelevant. First, because of the low expression of ATP synthase, and second, because of the high expression of UCP1 and the capacity to decrease membrane potential below a threshold in which ATP synthesis could take place. However, we discovered that IF1 is decreased upon 5 days of cold exposure in mice. And just to give a brief context, 5 days of cold exposure is enough to increase mitochondrial biogenesis and also increase UCP1 activation and expression. And what we found in this study was the downregulation of IF1 is a necessary process to allow the full capacity of mitochondria to adapt to cold exposure and to generate heat. And in this piece of ideas and speculations, we suggest alternative roles that IF1 could have in the adaptation of brown adipocytes during cold exposure. IF1 is classically described as an inhibitor of ATP synthase. When the membrane potential of mitochondria decreases to a certain threshold, IF1 is activated and binds to ATP synthase, stopping the reverse activity of this protein. And the regulation of ATP synthase by IF1 is very important in several different scenarios. For instance, ischemic reperfusion, cancer metabolism, and also in severe dysfunction of electron transport chain. However, recently studies in different contexts have pointed out IF1 as a master regulator of metabolism, cell fate, and mitochondrial morphology. In this piece, we speculated several avenues that are so far unknown when it comes to brown adipocytes and adaptation of cold exposure. For instance, we know the adaptation process during cold exposure increases the proliferation of pre-adipocytes into mature brown adipocytes, also increases mitochondrial biogenesis, and modulates the morphology of those mitochondria in brown adipocytes. However, we don't know if IF1 has a role in those processes. We're super excited about this new role of IF1 in brown adipocytes in adaptive thermogenesis, and we hope that our piece could stimulate researchers to test IF1 in different contexts other than regulation of ATP synthase.